So today I'll be talking about the paper Chemokine and FGF signaling act as opposing guidance cues in formation of the lateral line primordium. The study focuses on the collective cell migration on the posterior lateral line in zebrafish. The posterior lateral line arises from a cranial placode that forms sensory neurons and a primordium that migrates as a cohesive group towards the tail, depositing future neuromasts at intervals. The key findings of the study are as follows. It was shown that prior to the onset of migration, there are clusters of isolated cells called SPC or separate primordial cells caudal to the main primordium that express markers of the lateral line primordium. These cells are retained in position such that they fuse with the migrating primordium and contribute to the leading zone and later to the terminal neuromasts. It was shown that this retention of the isolated cells require FGF signaling that mediate attraction towards the primordium, thus contracting caudal chemoattraction by STF1 alpha. The formation of a single cohesive primordium is thus established by opposing chemoattractive signals that act on the initially isolated cell clusters. To understand better the events that precede the onset of migration, the study analyzed the expression of markers of the posterior lateral line placode and migrating primordium, HMX3, EYA1, and the Claudin LNGFP transgenic line, which very nicely marks the lateral line. This revealed the presence of one or more small clusters of cells expressing HMX3 that are located posterior to and clearly separate from the main body of the primordium between 10S and 26S. These clusters express the placodal marker EYA1 and are labeled by Claudin LNGFP. These cells thus have a lateral line primordium identity and they are referred to as separate primordial cells. SPCs were rarely seen after 24S, suggesting that they either die, fuse with the main primordium, or migrate to another location. No apoptotic cells were detected in this region, whereas a pattern of HMX3 expression suggested that SPCs are fusing with the primordium at 24S, supporting the coalescence hypothesis. To distinguish these possibilities unequivocally, the authors performed time-lapse imaging on Claudin LNGFP embryos. At 18S, the primordium could be identified as a large cluster of cells extending to somite 1. Time-lapse studies revealed the existence of a small group of GFP positive cells, which you can see here labeled with the white arrowheads, located caudal to the primordium. The primordium sequentially coalesced with these clusters and continued its migration, confirming the fusion hypothesis. Here is a movie of the time-lapse study. Initially, the primordium could be identified as a large cluster of cells extending to somite 1. We can also see small groups of GFP positive cells located caudal to the primordium. The primordium sequentially coalesced with these clusters and continued its migration, confirming the fusion hypothesis. Tracking analysis of the SPCs and the primordium tip cells indicated that the fusion is a consequence of the forward movement of the main primordium rather than backward movement of SPCs. When embryos were knocked down with the CXCR7B morpholino at 28 hours post-fertilization, 10% of CXCR7B morphins showed a split phenotype with a complete migration block and an SPC cluster caudal to the main primordium. This phenotype is interpreted as resulting from an absence of coalescence between the main primordium and SPCs. Directional caudal migration of the primordium is controlled by the guidance of CXCR4B and CXCR7B. As SPCs express markers of primordial cell identity, this raises the possibility that they too are subject to STF1 alpha mediated chemoattraction. SPCs express CXCR4B from 18S STF1 alpha expression in the somites start at the same stage, but CXCR7b is only detected at 26S in the trailing part of the primordium and is never seen in the location of SBCs. Here we have a high magnification view of CXCR4b expression blue in a Claudin LEN-GFP embryo, which marks the embryos in green. The study next addressed the effect of loss of function of CXCR4b on SBCs. The authors analyzed HMX3 expression at 24S in homozygous ODY mutants and stage matched controls, and calculated the frequency of SPCs defined as a proportion of embryo sites in which SPCs could be identified at this stage. And it was found that SPC frequency was significantly decreased in ODY mutants. Cell tracking revealed that SPCs move backward and fuse with the immobile main primordium. A simple model to explain these findings is that CXCR4b mediates a caudal chemoattraction of SPCs, probably through the sensing of STF1 alpha. However, this effect is normally counterbalanced by attraction towards the main primordium, which is revealed only when CXCR4b signaling is impaired. It is known that FGF signaling has a patterning role within the migrating primordium. The study analyzed the expression of FGF signaling components and found that after fusion between the SPCs and primordium, FGF3 and FGF10 are expressed in the leading zone and FGFR1 in the trailing zone. However, the expression pattern is strikingly different before fusion, when FGF3 and FGF10 are expressed in the back of and throughout the main primordium respectively, but barely detectable in SPCs, and the receptor FGFR1 is expressed in the main primordium and in SPCs. The authors hypothesized that FGF ligands produced by the main primordium could act as chemoattractive cues for the FGFR1 expressing SPCs, thus mediating the backward migration in CXCR4b deficient embryos. To test this hypothesis, the HSP70 TNFGFR1 line was utilized, which allows heat shock induced expression of a dominant negative form of FGFR1. Blocking FGFR activation from 16S led to a higher SPC frequency at 24S. Quantification of the distance covered by the tip of the main primordium and SPCs showed that the increased SPC frequency in HSP70 DNFGFR1 embryos is mainly due to a forward shift in SPC position rather than a defect in migration of the main primordium. To visualize the effects on the dynamic behavior of cells upon FGF signaling impairment, the authors used the FGFR inhibitor SU5402 in Claudin LEN-GFP embryos. Treated embryos showed an increase in SPC frequency at 24S, although milder than when the HSP70 DNFGFR line was used. Quantification of the distance traveled by the main primordium and SPCs at 24S revealed a forward shift in SPC position upon SU5402 treatment, with no significant change in main primordium migration at this stage, as also observed in HSP70 DNFGFR embryos. Live imaging of SU5402 treated embryos showed that SPCs underwent a forward movement before being eventually caught up by the main primordium. The study analyzed next whether the backward migration of SPCs observed in CXCR4b deficient embryos is mediated by the FGF pathway. To inactivate FGF signaling in CXCR4b deficient embryos, the authors used HSP70 DNFGFR1 embryos, which were injected with CXCR4b morpholino. Similar to ODY mutants, CXCR4b morphins displayed a reduced SPC frequency, confirming the efficiency and specificity of the CXCR4b morpholino. Blocking of FGFR activation prevented backward migration of SPCs seen in CXCR4b morphins. To investigate whether FGF ligands are sufficient to mediate SPC attraction, the authors transplanted FGF10 or controlled TD tomato expressing cells into wild-type hosts and analyzed the position of SPCs. species were found to have deviated towards ectopic dorsal sources of FGF10. Taken together, these results show that FGF signaling from the primordium attracts species, which by counterbalancing the caudal attraction to SDF1 alpha keeps them in place so that they fuse with the main primordium once it initiates migration. To analyze the contribution of species to the lateral line, the authors conducted fate mapping experiments using chiral photoconversion. Chiral expressing species were photoconverted from green to red before the fusion. In most embryos, red cells were found in the leading zone of the primordium, as you can see here, and later contributed to the last 3 neuromasts. The authors next performed laser ablation experiments. Partial ablation of species, as you can see in these white arrowheads, before the fusion led to impairment of the terminal phase of migration, consistent with the results of fate mapping. The primordium was smaller, stopped before the normal endpoint, and failed to split to form the terminal neuromasts. The formation and deposition of more anterior neuromasts was not affected, and the shape and organization appeared normal. Taken together, these data show that species are crucial for formation of the terminal neuromasts. The authors propose the following model. The balance between SDF1 alpha and FGF3 or FGFR1 signaling normally leads to an absence of anterior-posterior directional movement of species such that the migrating primordium meets and fuses with them. However, if species are unable to sense SDF1 alpha, they migrate back towards FGF-producing main primordium in an FGFR1 dependent manner. By contrast, compromising FGF signaling releases the species from FGF-mediated chemoattraction, resulting in the CXCR4B dependent short distance forward migration on the SDF1 alpha expressing path. Lack of CXCR7B in species prevents the long-term directional forward migration. These opposite movements reveal a tug of war between two guidance signals which maintain the position of species. In summary, the work reveals a novel role for the FGF pathway in chemoattraction mediated assembly of a migrating cell group in the posterior lateral line. It will be interesting to uncover whether similar chemotactic mechanisms could initiate or maintain a compact state of other migratory cell groups during normal development or cancer invasion. Thank you for your attention.